into different categories and narrow your differential diagnosis, especially if you don't have the luxury of doing a whole genome sequencing or West analysis, then you are limited by some genes. Now, the autosomal dominant ataxia, as you can see in this study where they looked at 55 patients, and it was found that about the spinocerebellar ataxia type 2 was the highest in prevalence in this study. Sorry, I'll go back. Yeah. So it's very important to look at the progression of the autosomal dominant ataxia patient, whether it is slow progression or rapid progression. Or if you've got a slow progression, then you have to look at the age of the patient. Having a patient less than 50 years of age, you think of certain spinocerebellar ataxias like SCAR1, SCAR2, and SCAR3. If the patient is more than 50 years, then you're thinking of probably SCAR6. If there's associated pigmentary loss, as I just said earlier, and visual loss, then SCAR7 probably would be the choice. Now, this is just a video of spinocerebellar ataxia type 2. This is a 55-year-old man with SCAR2 with limited upward gaze and slow saccadic eye movements and dysmetria. You can see the slow saccades. Some dysmetria here. There's pass pointing. So, what about geographic variation and prevalence of the ataxias? Well, scars usually are distributed in these countries, as you can see. SCAR6 usually is common in Japan, Australia, and Germany. SCAR3, which is the Mercado Joseph, more in Brazil and Germany and Japan and China, Italy and United Kingdom, SCAR1, UK, France, and USA, probably SCAR2, and Mexico and Brazil, SCAR10. Now, there's little actually data on the spectrum and frequency of the autosomal dominant scars from the African continent. However, there's one study in South Africa that pointed out that SCAR1 accounted for about 40.7%, followed by SCAR2, SCAR3, and SCAR6. You can see here, these are some published reports of the African families with inherited ataxias of scars types 1, 2, 3, 6, 7, or unidentified inheritance in some of the African countries. So this is Kenya. I'm not sure whether there are reports from Kenya on scars, but this is the limited information that I could find on the African continent. This is the same paper, actually. You can see here the distribution of the South African families diagnosed with scars type 1, 2, 3, 6, and 7 from 1990s until 2003. And you can see 50% of these patients had spinocerebellar 1, and 27% had SCAR7. And the ethnic distribution was mainly unknown, but there were lots of Black African, about 36%, and of mixed ancestry as well. So moving on to talking about the distinguishing clinical features in addition to ataxia, pyramidal signs, neuropathy, actually points you towards a diagnosis of SCAR1. Slow saccades, neuropathy, hyperreflexia, and dementia points towards SCAR2. A slowly progressive disease towards SCAR5. You've got Parkinsonism, which is a prominent feature of SCAR3. Visual loss, as we pointed out earlier, due to retinal pigmentation, identifies SCAR7, and then a and so on. So it's important to look at the different features. For example, if you've got a patient with significant dementia, Parkinsonism, dystonia, and psychiatric or chorea, or Huntington disease-like disease, or seizures, then you're probably looking at SCAR17. So it's important to look at non-ataxia signs and symptoms, suggesting specific genetic subtypes of autosomal dominant spinocerebellar ataxias. And as I just pointed out, pyramidal features are important to look for motor neuron involvement, peripheral neuropathy, slow saccades, ophthalmoplegia, eyelid contraction, macular degeneration, tremor, Parkinsonism, dystonia, myoclonus, and each of these features just points you towards a different type of spinocerebellar ataxia. Now, one of the common autosomal dominant ataxias associated with repeat expansions, as you can see in this table, includes SCAR1, 2, 3, 6, 7, 8, 10, and 17. And it's important also to look at for the associated clinical features I've pointed out before. And now SCAR-A1 is characterized by dysphagia, dysarthria, pyramidal signs, and SCAR2 with slow saccades. And again, the visual loss, which I stress on, comes more with the SCAR7. Now, spinocerebellar ataxias do masquerade as something else. So sometimes you'd have a patient with specific focal dystonias or task-specific focal dystonias, and you may think this patient has got actually a dystonia and treat him as such. But in fact, SCAR7, 6, and 17 can present as such, and then later on, probably the symptoms may unreveal on the signs. Some present as young onset dopa responsive Parkinsonism, such as the SCAR3 and the SCAR2. Some of the scars present also as Huntington-like disease, like SCAR17. So there's no one-to-one relationship between genotype and phenotype. Now, SCAR2 is a great mimicker, and it's important that you note the different or variable phenotypes of SCAR2, because it depends actually on the CAG repeat expansions. If you've got a repeat expansion of more than 40, then the ataxia will be more prominent. You have the phenotype with ataxia. If there's a lower repeat expansion of 34 to 39, you probably have the Parkinsonism phenotype, and the ALS phenotype with a more lower repeat expansion of 32 to 33 CAG repeats. It is a variable age of onset. It's not dependent on the number of CAG repeats, but as I said, it's just the different phenotypes with the different CAG repeats. There's earlier disease onset, the more severe symptoms. SCAR3, which is the Mercado-Joseph disease, very prominent with Parkinsonism, and the gene is ATXN3, and the clinical phenotypes include ataxia, dystonia of the rigidity, Parkinsonian, and amyotrophy as well. And there are different subtypes. The type 1 disease is of younger onset, and it's characterized by spasticity, rigidity, bradykinesia, but minimal ataxia. The type 2 is characterized by spastic paraplegia, so more of upper motor neuron signs, alongside with the ataxia. And the type 3, there's initially a peripheral polyneuropathy, and then at a later age, the ataxia comes on. The type 4 is very similar to dopa-responsive Parkinsonism, and the significant feature is Parkinsonism. The second condition that I'd like to discuss, obviously, we cannot discuss all the conditions, but I've just pointed out the most important ones. The episodic ataxia type 2 is characterized by the attacks of ataxia, dysarthria, nausea, vertigo, diplopia, oscillopsia lasting minutes to days. And there's interictal nystagmus or mild ataxia, and normally this is provoked by stress and exercise, but not startle. Attacks may respond very well to acetazolamide, and it carries the same gene as SCAR6, which is the CACNA-A1 gene, and this is also present in the familial hemiplegic migraine, and overlaps may occur around these conditions. So episodic ataxia type 2, SCAR6, and hemiplegic migraine. Patients may also have weakness with neuromuscular junction abnormalities. So what are the potential treatments for the spinocerebellar ataxias or autosomal dominant ones? Now, mainly the treatment is symptomatic, which I'll discuss at the end, but these are some of the potential treatments that studies are looking at, like the antisense oligonucleotides, which is a small single-stranded sequence of DNA that have gene targeting effect for reducing levels of toxic protein or making non-toxic modifications. The RNA-based therapy, gene expression silencing, and stem cell-based therapy. Moving on to speak about the autosomal recessive ataxia, and these are categorized into three types: the cerebellar ataxia with predominant sensory neuronopathy, cerebellar ataxia with sensory motor axonal neuropathy, and cerebellar ataxia without sensory neuropathy. And the clinical classification actually overlaps. The gene associated with each primary recessive ataxia is classified according to the most frequent clinical syndrome described for this disorder. And you can see that some disorders have more complex or variable phenotypes and are placed in the overlapping areas, as you can see here between the two categories. And the genes are actually presenting in large fonts represent the most prevalent ataxias out of these autosomal recessive ataxias. Again, the classic phenotype for autosomal recessive ataxia is the Friedreich's ataxia, and this is by far the commonest one. And it's important that you identify as soon as you get a patient that you think may have a spinal recessive type to identify whether this is present or not, before you go on to do multi-gene level panel or whole exome sequences, just to save money. So the autosomal recessive ataxias, as you can see in the study of 58 patients, the most prevalent, as I said, or the commonest one is Friedreich ataxia out of all of these. So it's important that you look at in the recessive patients for this type of ataxia before you go on to do some genetic testing. Now, commonest causes of autosomal recessive ataxia, as I said, the Friedreich's ataxia, and the genetic mutation is repeat expansions in the intron 1 of FXN gene, and the age of onset is usually from childhood to the third decade, and the associated clinical features include piscavus, scoliosis, hyporeflexia, and spasticity. There are other types, such as cerebellar ataxia with sensory motor axonal neuropathy, such as ataxia telangiectasia, ataxia with oculomotor apraxia type 1 and type 2, and the cerebrotendinous xanthomatosis. Those without sensory neuropathy include the autosomal recessive cerebellar ataxia type 1 and type 2, and the Niemann-Pick disease. I've just put this up because I found that this is a very nice review so the first one is the systematic review of autosomal recessive ataxia and their classification, and this you can access through the movement disorder website. It's the genetic nomenclature of recessive cerebellar ataxia, and I found this very useful in preparing this lecture. So which autosomal recessive cerebellar ataxia do you consider based on the prominent features? Well, if you've got the peripheral neuropathy, then you're probably looking at ataxia with oculomotor apraxia, either type 1 or type 2. If there is a significant oculomotor apraxia, then ataxia probably telangiectasia, or either of the previous, the oculomotor apraxia type 1 and 2. Retinitis pigmentosa also. You can find out which type of ataxia there is, oculomotor telangiectasia as well, ataxia telangiectasia, dystonia, you're looking also again at ataxia telangiectasia, and so on. So it's important to see the cerebellar features or the prominent features alongside the ataxia. Now, again, as I mentioned, there's not a specific medication that treats neither autosomal dominant or the recessive ataxia. However, there are some symptomatic therapies and some therapies that are in the pipeline and have been looked at. For example, in autosomal recessive ataxias, the disease-modifying therapies, for example, Glucocorticoids have been tried in ataxia telangiectasia. Probably it restores the ATM gene expressions. Others, such as chenodeoxycholic acid supplementation, so some supplements replacing the missing bile ducts and lipid metabolism deficit as well, replacing ataxia with vitamin E deficiency with vitamin E, and autosomal recessive cerebellar ataxia type 2, again with replacement therapy, and so on. So not a specific therapy, but I would say some therapies that probably help. So just going again over some of the autosomal recessive ataxia which are common. Friedreich's ataxia, as I said, that involves the FXN gene, and it's characterized by decreased proprioception, areflexia, squaroid jerk, scoliosis, and high arch feet. So look for these features as well. Ataxia with oculomotor apraxia. Look for oculomotor apraxia, so this is the first thing that you look for. Telangiectasias in the eyes and skin, especially of little children. You would think of ataxia telangiectasia. Pure ataxia, you'll think of the autosomal recessive cerebellar ataxia type 1. So if you don't find any other features, then probably this is what you're looking at. So just a few slides on Friedreich's ataxia, because it's common. The prevalence is 1 in 50,000 persons. I would say, so when I say common, it's because it's the commonest out of the autosomal recessive. And the age of onset is generally before 20 years, and it's characterized by ataxia, dysarthria, sensory loss, corticospinal tract signs with absent reflexes. So it's important to examine the patient thoroughly, not just for cerebellar signs, but also from accompanying signs like peripheral neuropathy signs. Skeletal, look at this skeletal abnormalities for kyphoscoliosis. Look at other systemic features and systemic examination, whether the patient's got diabetes or cardiomyopathy as well. The genetic abnormalities in expanded trinucleotide repeat of GAA in the first intron protein coding for the protein called frataxin, and 6% of the time there's a point mutation. This is a mitochondrial protein encoded by nuclear DNA. It's involved in ion transport, and its abnormality leads to mitochondrial dysfunction. You can see here the thinning of the spinal cord, and this is one of the MRI features of Friedreich's ataxia. Another one, as you can see here pointed by the arrows, is the symmetric increase in signal within the middle cerebellar peduncle. I've just brought in this slide to show you the conjunctival telangiectasias in a patient with ataxia telangiectasia, and this can also be present on sun-exposed areas, especially in children. Now, some of the ataxia present at an older age, especially the condition called canvas, and patients normally have cerebellar ataxia, neuropathy, vestibular areflexia syndrome, and can have chronic cough, and this is related to a RFC1 repeat expansion, and this is a common cause of late onset ataxia. Moving on to talk about the X-linked ataxia, and here I talk about the fragile X-tremor ataxia syndrome on the X chromosome, and the gene here is FMR1 gene with pre-mutations with 55 to 200 CGG repeats, and carriers in males range from 1 in 813 and 1 in 259 in females. Females usually have premature ovarian failure with menopause less than 40 years. The clinical symptoms include late onset ataxia, more than 45 years of age, with action tremor similar to essential tremor, and with postural component being more than the intention type. Ataxia is usually axial and appendicular. There's Parkinsonism as well, a peripheral neuropathy exists, and autonomic symptoms as well as cognitive changes. I will show you the MRI features of this condition, and you can see here on the left axial flare with white matter hyperintensities and global atrophy. On the right, you can see the axial flare with hyperintensity in the middle cerebellar peduncle. And moving just one or two slides on mitochondrial ataxias, and these are mitochondrial DNA mutation that can cause ataxia as well. And these include Kearns-Sayre syndrome, myoclonic epilepsy with ragged red fibers or MRF, neuropathy, ataxia, and retinitis pigmentosa, the POLG-related disorders, which includes ataxia, neuropathy spectrum, and mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes called MLAS. And the diagnosis is usually by muscle biopsy because mitochondrial defects are more likely to be detected in the non-dividing cells. And here you can see a graphical summary of the clinical approach to a patient presenting with ataxia, regardless of the type. So most important, you must obtain a clinical history with temporal course, family history, physical examination, and paraclinical tests, and then exclude all the acquired causes, move on to evaluate the mode of inheritance, looking and digging more into the family history, and evaluate the clinical syndrome, and to assess whether it corresponds well to a characterized genetic disorder or not. So mainly looking at the associated signs and symptoms, the age of onset, and digging more into the family history. Test for the commoner ones, the Friedreich's ataxia and other specific single genes, if high clinical suspicions or confirmed mutation is present in a relative, and then test for multiple genes through next generation sequences if you've got the luxury to do that. So just some clues on what genetic conditions out of all the ataxias to look for or to suspect. If you've got a patient over the age of 50, then you're probably looking at SCAR6. And here you probably need to check for the CACNA1 gene, and you might need to inquire more about the overlapping conditions here, such as the familial hemiplegic migraine as well. Before age, also episodic ataxia with SCAR6. Before age 20, you're probably looking at Friedreich's ataxia or ataxia with oculomotor apraxia types 1 and 2. If it is a younger child, before the age of 5, probably ataxia telangiectasia, so look out for those telangiectatic lesions, whether in the eyes or sun-exposed areas as well in the skin. And this actually subjects them to more high risk of cancer. In infants, look for SCAR2 and SCAR7. Also look at the associated features, as we've discussed before. Retinopathy is very common with SCAR7. Seizures is common with SCAR10, and dementia of SCAR2 and SCAR17. Now, if you're heading towards a genetic confirmation of these conditions, and you can't check all the genes, obviously, because of the cost, then you check the commonest ones. Out of the autosomal dominant, the commonest are SCAR1, 2, 3, and 6. Out of the recessive is Friedreich's ataxia and fragile X-tremor ataxia syndrome out of the X-linked ataxia. So there's limitation of actually of whole exome sequencing, which is not sensitive for large chromosomal deletions or duplications, such as in SCAR20 or for ataxia associated with mitochondrial genome mutations or for repeat expansions in the non-coding regions. So we still have some limitations in the whole exome sequencing. In this slide, you can just see the different medications that are used in terms of symptomatic therapy for some of the features that are associated with ataxia. For example, if you go a patient with spasticity alongside with ataxia, then you could try baclofen, tizanidine, or dantrolene, or botulinum toxin injection, whatever is available in your country. If there's associated tremor, then probably propranolol or primidone topiramate. Dystonia can respond very well to botulinum toxin injections. And fatigue, you can just check for probably sleep hygiene or physiotherapy in general. Antidepressant can work well if depression is a symptom. So really depending on the symptom that you have or the signs that you have in these symptoms, then you can probably guide your symptomatic therapy as such. So, but generally speaking, physiotherapy, occupational therapy, speech and language, palliative care, so a multidisciplinary approach is very important in patients with ataxias. This is a comprehensive systemic review summary actually of treatment of cerebellar motor dysfunction and ataxia. And here for patients with ataxia of mixed etiology, probably riluzole improves ataxia signs at 8 weeks. Patients with Friedreich's ataxia or spinocerebellar ataxias, again riluzole improved ataxia in this study at 12 months. Additionally, valproic acid has been tried in patients with spinocerebellar ataxia type 3, and it improves the ataxia at 12 weeks. Those patients with spinocerebellar degeneration or thyroglobulin releasing hormone possibly improve some ataxia signs over 10 to 14 days. So that should be tried as well. In conclusion, there are many types of ataxias that are still not identified, and determining the etiology of cerebellar ataxia is complex. A step-by-step approach can streamline the diagnostic overflow. The spinocerebellar ataxia nomenclature relates to the autosomal dominant causes of ataxia mainly, and the first approach to the genetic of ataxia is to investigate for repeat expansions and look for those common ones first. Etiologic therapy is uncommon, but sometimes can be done. For example, if you're replacing a therapy or gene silencing, etc. And these are still being studied. Symptomatic therapy, although unsatisfactory in ataxia, but it helps with the other accompanying symptoms. Thank you very much.
Thank you very much for your nice presentation. And we now take questions, comments from the audience. Anyone has a question? Okay, let me just ask Dr. Zakaria. Can you hear me?
Can you repeat that question again, please?
Oh, okay. No, I'm about to ask the question. So,
sorry?
Yes, are there mutation-specific database or databases for ataxia, hereditary ataxia?
I'm sorry, the line is cutting. Can you probably write it for me in the chat?
Are there mutation-specific databases for hereditary ataxia?
Database for hereditary ataxia, is that your question?
Yes, correct.
Yeah, I'm not aware of any.
Okay. All right. So it might be interesting to look into that in the future to have mutation-specific databases for hereditary ataxia, so that young investigators or any career investigators who might be interested in studying hereditary ataxia could have something to start with by genotyping those mutations in their patient. So it might be something to look into in the future.
Yeah. Actually, I do have questions, for anyone. Just preparing for my talk, I didn't find so much information on hereditary ataxia in Africa. I don't know which is the commonest. I mean, I've found two studies that I've just showed in my presentation, but which is the commonest spinocerebellar ataxia that you see in the African countries?
Okay, thank you very much. Does anyone have a question again? Okay, please a round of applause for our presenter, please. Thank you, Dr. For that presentation. You can also stay, Dr. Zakar. You can also stay online for other presentation. Okay, so we are getting there. Before we go to the next talk, I think I'm the next presenter, and luckily I've made the slide. Just, I think maybe 10 or 9 slides. So that will be very quick, and then we can go for our tea break. But before we go there, I would like to ask, throw this question to the students that are accepted for this Igbo school. I would like someone to stand up and tell us what are the key highlights of what you have learned so far since we started in the morning. So someone to just stand up and tell us important key highlights that you have noted and benefited before we go to the next presentation. Who wants to talk? Are you a student of faculty? Okay, good, good.
We're all here to learn. Well, I cannot use my non-stick pans anymore to protect myself against Parkinson's disease and those in my house. So that was something I got to learn today.
And who else, any volunteer that want to give us, at least to tell us what you have benefited some from, the knowledge that has been added to your knowledge before this workshop started this evening. Okay, madam.
What I learned this morning is about mutation in Parkinson's disease. That was, Zurike will say that the mutation is are influenced by ethnicity and geographical situation. That was I learned. It was very interesting for me.
Fantastic. I mean, that's important. Can we give her also a round of applause? Okay. So now we can go to the next presentation. And like I said, I've made it so brief so that we can quickly go for our tea break and then go straight to the student presentation. Yes, the second presentation. So just 10 slides, and you can please project it. No, start from the first one. Yeah. I was just saying that it's going to be a quick one. Okay, so I've been asked to talk about the neurogenetics of the cerebral cortex, phenotyping and mutational analysis. So, but like I said, I believe that I'm talking to neuroscientists. This is a neuroscience conference, so I will not be going into details of cerebral cortex. All of us know what that is, and then the most important thing is that I'm interested in because I look at it that if you look at it in terms of landscape, most of the diseases that we develop, neurological diseases, are centered in the area of the cerebral cortex. Talk about Parkinson's disease, Alzheimer's, and so on and so forth. So it might be interesting to understand the phenotyping and mutational analysis in this aspect. So the protocols include gene mapping and identification of mutations. And then we talk about network base for relationship among genes and various phenotypes. And then we talk about important loci and common single nucleotide polymorphism cluster identification in this brain area called cerebral cortex. And there's a lot of biological activities going on in the cerebral cortex, isn't it? So that we are aware of. So this is one of the important studies by Ayasaka et al. And Kramer et al. And then what they have done so far was, and they are leading the way to tell us in terms of gene mapping, the diseases, the brain area, and then the common diseases. So this is a model that shows us that they are mapping the brain area with various neurological diseases, and then they were getting common diseases with brain area, and then again it moved on to another model that shows the brain area with common diseases, and then at the center of them genes regulating the certain brain area and then disease. And that also leads to that question that the gentleman asked me earlier on about dopaminergic neuron, where some areas are affected and some area might not be affected. And the answer is actually based on gene mapping that shows the area. And then in that study, they've been able to establish more than 500 genetic disorders associated with about 2,000 nodes, genetic nodes in the brain area, and most of them emerge from the cerebral cortex. And that is kind of important for any genetic diseases that are related to a neurological disorder. So we are talking about the most active region in the brain, cerebral cortex, for some of these neurological diseases, and it is important to pick up those mutations in that area. And then another protocol will be the hereditary pattern. It is important to study the hereditary pattern of these diseases in the cerebral cortex. So it is about determining the mode of inheritance. We have heard about autosomal recessive, autosomal dominant, and mitochondria. And these three are actually the most important phenomena when it comes to discovering genetic mutation and environmental factor related to any neurological diseases or most of the neurological diseases that are inherited. In this autosomal recessive, and most of the genes are actually inherited in autosomal recessive pattern. Like we can see here that autosomal recessive, we have a situation whereby the father is a carrier, then the mother is also a carrier, and then they are not showing the phenotype. They are just a carrier because there's a dominant trait that overshadow the phenotype. And then during genetic courses, when they start giving birth, we could see another two carriers here that are not showing the phenotype. And then we can see another child without being a carrier or showing the phenotype. And then we will see another child that shows the phenotype. And then they might be wondering that, like in Parkinson's disease and also hearing impairment that I was studying in Cameroon, we saw a lot of questions in Ghana, a lot of questions whereby parents were not having hearing impairment, and then the children, or one of the children, developed hearing impairment, and then the man abandoned the woman and ran away and said, "Oh, such is not in my generation. It must have come from your own generation," and and then it left a lot of controversy. And the only simple explanation for that is that the man actually is a carrier, and then the woman also a carrier, and then it surfaces in their children. And one of the phenomenal important thing about autosomal recessive, it is very very controversial as it is because it skips generation. So it might not occur in this generation. All of the children can even be a carrier. Then the next generation can even still be a carrier. Then one case will just surface, and then they will say, where did they come from? It's probably not our child, or it's probably not in our line. Whereas they have been a carrier all over so many generations. And most of these Parkinson's disease genes are inherited in autosomal recessive manner. And that is why it is difficult to have genetic diagnosis because the people are not developing the phenotype, or they have not even lived enough to develop Parkinson's disease at old age. So then the one for autosomal dominant, it is very obvious. It is contrary to autosomal recessive because every generation will show at least there will be a case because it will be dominant case, and during genetic courses, either the child will have it, or either the boy, a male child or a female child will have it. And then another phenomenon is the mitochondria, which is, we all know that we inherit mitochondria mostly from our parent, only from our mother, mitochondrial DNA from our mother. I don't know why God made it like that only the female have mitochondrial DNA to transfer to the offspring, but men, there's an explanation for that. If you want me to say it, I can explain why, but God made it like that. So then the children are affected because the mother has transferred the mutated mitochondrial DNA to them, and mitochondrial DNA is one of the crucial steps that people have been using to understand population genetics, because it goes from, because you can track it to just a family, a female, and then it goes on, goes on, goes on like that, because it must be inherited. So then mutational analysis, we can have a flow of how we can, after we have collected sample from the patient, DNA has been extracted, genotyping has been done, and then we generate FASTQ data. We call it FASTQ data. And then after generating the FASTQ data, we will do what we call mapping alignment. So the mapping alignment is what you do with the reference genome with the Human Genome Project that I talked about earlier on. And then after mapping, you do positional sorting, and then you do duplicate marking, whereby so many duplicates, you have to remove them. You trim them out using a lot of software, which I'll be explaining later on. And then we do what we call variant calling. So we want to call the variant from the data, and then, like I said, they are just genetic codes. Then we, after doing the variant calling, then we can now have what we call joint VCF, GVCF. So we can now have a GVCF that will now be used for joint calling, joint genotyping for a final VCF. And what genotyping, joint genotyping means is that you are bringing in all the cases together, maybe a family, and then you are bringing them together to do a joint call of what are the things that are inherited together in this family, and what are the things that are not inherited. So when you do that joint calling, you will see some things that are not within the family, then those ones you can refer to them as mutation, that is within the same family. So then there is a pipeline for this, a lot of pipeline, bioinformatics pipeline. According to Jennifer, until 2016, some of these have been updated recently. You talk about the alignment. So this is whole exome sequencing data, the FASTQ data that I'm talking about. So it comes like a binary form. You can open it usually using your Unix, Linux system, Ubuntu, and so on and so forth. And then there are tools that you can use for various analysis, SAM tools. You can use PCAD, you can use FastUnit, and then for the alignment. These are programming languages that can be used. You can also use BWA, and then the bowtie. You can use all these tools, which have been invented by many smart bioinformaticians. The joint calling, you can use example free bayes. You can use GATK, which is mostly used. SAM tools as well have been mostly used for joint calling that I explained. And then the variant calling validation, you can use ANOVA. For example, you want to know what are the pathogenicity level of the variant that have been identified, annotation, we call it variant annotation. And you can compare this with 1000 Genome Project, DB-SNPs, that is for database for single nucleotide polymorphism, and so on and so forth. And then we can do so many analyses, including checking what are the different proteins that are being expressed, copy number variation in the sample. Copy number variation is when you have more copies of genes than the other, and that you can explain why Down syndrome is a genetic disorder, because you have extra chromosome that causes it. And then after that, you can do ClinVar analysis, and then you can do FAMGKB. This is actually for pharmacogenomics analysis, and then you can do you can check the drug bank database to see the relevance of this mutation in clinical cases if they have been previously reported. If they have not been previously reported, then there's a lot of work to do. One of them is to, first of all, prioritize the variant according to the guidelines by the expert. This was a guidelines by the expert that was published as a perspective And it has been guiding most of us in this field to know about how to investigate causality of sequence variant in human diseases. So there are guidelines, like these are the guidelines, the terms used to describe sequence variant. So one of them is that pathogenicity. So what is actually pathogenic? It contributes mechanistically to disease, but it is not necessarily fully penetrant. That is, may not be sufficient in isolation to cause the disease. So what is implicated? Possesses evidence consistent with the pathogenic role with a defined level of confidence. And then what is associated? Significantly enriched in disease cause cases compared to match control. You know, those are the things I was explaining earlier. Damaging, it alters the normal level of a biological function of a gene or gene product. Or deleterious, it reduces the reproductive fitness of carrier and will be thus be targeted by purifying natural selection. So this, once one of the variants or mutation falls into this category, then we can begin to prioritize either of these variants after the analysis for important future study. And one of the future study will be to do molecular study like wet lab, gene cloning. So gene cloning, the roles of mutation in genes can be studied functionally with gene cloning. And it is actually a fine approach to study the function of a gene in a relatively stable condition. That is, in a lab, most of us will have heard about molecular cloning, whereby you construct, you open up the vector, bacterial vector, and then you insert a construct that includes your DNA mutagenesis. And then once you do that, you do your ligation. Once you do your ligation, because you have cut the vector with restriction enzymes specific to each site, and then you do your ligation, and then you digest, and then ligation. And once that perfect construct is already achieved, that has the mutation in, you can begin to clone this in bacterial vectors to produce more copies. You can also do PCR to produce PCR polymerase chain reaction to produce many copies of this. And then once you have achieved that, the next thing is to do gene expression assay. So you can look at the expression level of the gene versus a mutant versus non-mutant, like this control cases. I'm sure you will have seen a lot of this in many publications in neuroscience, whereby they run gels, Western blot, and then they will show you expression level in mutational cases versus control. They are trying to compare the gene expression level in these cases. And then you can also do what we call COS assay, and that is for mitochondria stress level in terms of disease that affect the function of the mutation in the disease cases versus the control cases. So, and again, you can also do in silico analysis using protein modeling. This was one of the my study and my students at UCT that is currently given full consideration for publication at the scientific report, Nature. And then what we did a lot of protein modeling that can infer into what the effect of mutation in proteins function. So there are many tools, many in silico tools to start with. Once you understand that, okay, this mutation can cause a disease or can be associated with a disease, then you can go into the knowledge to say that, okay, what are the tools available to model a reference gene that is not mutated versus a mutated gene. And then once you do that, so like this in this study, we did a control and then a mutant. And then we can see how the deleterious level was moving towards in the disease control. And then it affects the 3D dimensional model of this protein. And also it affects the hydrophobicity of the protein in this particular region compared to a wild type. So hydrophobicity is talking about water loving, isn't it? That is, the cell protein begin to absorb more water compared to the one that is not mutated. And lastly, this also was in that study. You can do what we call comparative genomics, whereby the mutation, you can look at it, is it actually a conserved protein in many species? And if it is a conserved protein in many species, like in primate, in mouse, in rodent, in fishes, and also all sorts of animals. And then if that protein is highly conserved, then it means it is an important protein, and any mutation in there will lead to genetic mutation. So thank you very much. That is a presentation.
Thank you, Femi, for this stimulating. Yes, Mark.
University of Ottawa. You showed a slide where you had the different adjectives to describe the impact of the mutation. Can you tell us how do you define? Are you able to determine that this is this particular category and not this other particular? What are approaches that can be used?
So determining, like now, thank you very much for that question. So like once we are doing the mutational analysis, so we get all sorts of mutation. So it could be copy number variations, it could be single nucleotide variation, it could be missense, and so on and so forth. But the first thing we do is that we look at first the location in the genome where they identify. If it is intronic, that is in the introns of the genome, there is a possibility that it might not affect protein expression, because we know that it cannot be transcribed into, it might not be transcribed into protein synthesis. So then if it is a missense mutation, that is, it alters protein synthesis, then we can begin to prioritize such. We can prioritize, that is non-synonymous mutation. We can prioritize copy number variation. We can prioritize other form of mutation that might impact the gene structure or the gene function. And then after that, we go into looking at the minor allele frequency of this variant. In my first lecture, I talked about susceptibility to disease, that at least a common variant will exist like more than 5% of the population. So if it's in order for, if it is having a minor allele frequency of about 5% or more than 5%, there's a possibility that it is not the mutation that is causing the disease, because we should have other people developing that have that same mutation developing the phenotype. So then we are now looking for rare variants that is in maybe 0.01% of the population, and then we can prioritize such variants for further analysis. And then after that, we can then look into databases to look at what have been reported about that mutation. Is it a novel one, or has it been associated with a disease before, or a phenotype that we are studying? So if there's no other information about that, then we can talk about doing what we call GWAS, that's genome-wide association study. But that will require a lot of power calculation, because it will require a lot of having sample to come to associating that variant to a particular phenotype of disease. Thank you.
Thank you.
Thank you.
Thank you so much, and I wouldn't want to get into the sciences that you are in. It's It's a complicated one, but I just wanted to comment on mitochondria inheritance. Although it was largely believed, and we had been taught in our biologies, that mitochondria can only be inherited from mothers, a lab in China, Tao Chen Huang's lab, published a paper in 2018 that describes that it is now possible, it is certainly possible to inherit mitochondria from both the father and the mother. So there is a biparental mitochondrial inheritance, and therefore a possibility of inheriting the mitochondrial disease from the father as well.
Just to respond on that, I think I also heard about that publication in China. So, but it has not received a famous acknowledgement. There have been a lot of backlashes to reject that idea, because sometimes what we know, the knowledge that we know is that the reason why the mitochondrial DNA are not inherited from the father is because in the spermatozoa, it is in the tail that the mitochondria is. Understand? Whereas in the ovary, it is compact in the ovary. So during conception, the fusion, you know, the head of the spermatozoa has to go, and then the tail has to go down, go away. So when it is cut off, the mitochondria is no longer there. You understand? So then it picks up the mitochondria of the mother for the conception to take place. So we cannot have two mitochondria. But that study has been receiving a lot of backlashes. And one of the possibility could be, you know, these days there is now this another means of conception that they call IVU or IVF. Yes, IVF. So, which directly introduces cell, that means sperm cell, into the ovary, and that alone maybe could lead to why their publication was accepted or given preferences in the first place.
Thank you so much for your presentations. While you were giving your presentation, I observed you were using the word disease and then the word disorder interchangeably. So I want to ask, are the two terms the same? While giving presentation, is it safe to use the two words interchangeably?
That is a very important question. Thank you very much. And I will say that, number one, I'm not a medical doctor. So medical doctor could also understand what I'm about to say. So disorder could have many diseases causing to forming a disorder, and then you could have a particular disease that you are talking about. So when we talk about Parkinson's disease, it could be just addressing Parkinson's disease. But when we talk about disorders, it could be many diseases that are forming a disorder. So, but I'm sure we have young medical doctors here, or experts who can maybe shed more light. My friend over there, Dr. Michael, do you have any other alternative answer to that? Perfect. Thank you very much. I hope that answers your question.
Next, in economies of silence, means do a cup of coffee, and then we go into the student presentations. I'm informed that majority of you are saying they're not ready. That I will accept. But then we had presentations for the next 3 days, and we only have 2. Means tomorrow Video presentations will have to be handed to you. It's up to you to decide whether you understand it or whether it is ready. Thank you. And can we do yes, there are two that are ready